Okay, so now I'd like to introduce Veera Baladandayuthapani. How did I do, Veera? He's the director of the Cancer Diet Data Science Shared Resource, and Gen Lee, assistant director for Cancer Diet Data Science Shared Resource.
Thanks, Evan, and your team for organizing this and giving us a platform to talk about cancer data science shared resource. I'll tell you a bit about who we are and how do we are taking a few steps in the directions of doing spatial analysis, in broadly speaking. So as Evan said, I direct the CDS over here, and Dr. Ganley is my colleague who is an assistant director for the bioinformatics operations. So overall, in the fabric of Rogel Cancer Center, where we sort of fit in is we are a group of biostatisticians, bioinformaticians who provide high level of analysis support to a spectrum of endeavors, research endeavors at the Cancer Center, and support a plethora of ongoing and developing research. And as I mentioned, statistical bioinformatics and informatics support. And a lot of us are, have research programs in developing advanced methods. And that's particularly germane when we get to the spatial analysis part, that we develop a lot of advanced modeling techniques for such data, and all with some anchoring in rigor and reproducibility. So overall, the structure of the core, as I mentioned, Evan's our fearless leader as the associate director of the shared resources, along with Brahmar Mukherjee. And we have lots of faculty with varying degrees of expertise, as well as a stellar group of analysts who work on different aspects of this. And coming to the single cell and spatial biology expertise, we are looking at in the broadest possible sense. Of course, the single cell, where one can do cell type identification and trajectory lineage analysis, just some flavors of types of analysis projects that we've been involved in. Spatial transcriptomics, of course, with the whole transcriptome and spatial heterogeneity, and understanding spatial heterogeneity, especially in the context of cancer. Spatial proteomics is sort of has been a newish kind of an endeavor. Looking at the different functional phenotypic state of the cells, and also just going way back and just the start of what might be spatial biology is through the histopathology, mining of these whole slide images, and looking at morphology of tumors. So as I mentioned, we have a lot of faculty and staff, and some of whom are here as well, who work on different aspects of this. I'll hand it over to Gen to talk about the single cell and the spatial transcriptomics. I'll jump back in for the proteomics part.
Everyone. So I'll give a little bit more details about the type of analysis and techniques that we can provide at the core. So the first one that we do is a lot of single cell RNA-seq data analysis. So for this type of data analysis, we typically take your data, which can be represented as a data matrix expression profile, and we conduct or develop these pipelines to go through the quality control, try to get rid of these low quality cells, genes, and before we do any additional analysis. So we also can perform normalization of the data so that these different cell sizes, different sequencing depths can be properly dealt with. And then dimension reduction, batch correction, those are the common techniques that we apply to the data so that we ensure that the data quality is high enough for any subsequent analysis. And this we can also use cluster analysis to identify the cell types, annotate the cell types before we do more in-depth analysis, such as the visualization using this HuMap or t-SNE that you are probably familiar with. And we can also do some standard tasks like differential expression analysis, cell annotation, etc. And on top of that, we can also take that one step further to conduct more in-depth downstream analysis. So here is an overview of the type of analysis that we can do at the core, including the pathway analysis, where we highlight and emphasize those important pathways that are sort of upregulated or downregulated in your processes. and also do this trajectory analysis, velocity analysis by assigning some pseudo time stamps to the cells, to individual cells, and to identify this trajectory in the pseudo time domain, and which can also be validated using lineage tracing technologies. We have done quite a bit of the actual analysis in all these aspects. I won't go into the details, but just to give you a overview, we've done the single cell analysis for prostate cancer samples in mouse. We've done it in kidney, renal clear cell carcinoma, in multiple myeloma, etc. But all these techniques can be easily applied to other non-cancer data as well. So if you have requests, single cell data analysis requests, feel free to get into contact with our core. Another area that we have strong expertise is the spatial transcriptomics. So compared to the single cell RNA sequencing data, so spatial add additional layer of complexity, but as well as the opportunity to the analysis. So it's as the previous speaker have already demonstrated, it's a rapidly developing area with a lot of challenges, but also opportunities. So in terms of the data analysis, in addition to processing this RNA sequencing data, we also have to deal with the imaging data as well. So we also provide these pipelines of analyzing the image data, including the image registration, alignment, as well as the cell segregation, so that you can overlay your image with your sequencing data to provide a more in-depth, comprehensive understanding of the data at hand. And once the data has gone through this quality control, the pipeline, again, a lot of downstream analysis can be performed. And I want to mention that another strength of the cancer data science core is that we actually house in the biostat department. And we also can borrow expertise from our colleagues from the biostat department, where we have a lot of colleagues actually developing these new methods and tools for spatial transcriptomics analysis. I'll give you a little bit of flavor in the next couple of slides. So the one example is the one of our colleagues has recently developed this new computational methods that can actually identify the cell type specific spatially variable genes. So we look at these spatially resolved transcriptomics data and use this spatially variant coefficient model to look at which genes actually have this spatially variable expression in the whole slide, and be able to identify the genes that present this spatially variability in the spatial domain. And we also have developed this very powerful, very comprehensive server, the spatially resolved transcriptomic server, the SRT server, which is essentially a web service that can provide a lot of standard analysis for spatial transcriptomics data. It doesn't require a lot of expertise in quantitative analysis. It's web-based, so you can easily upload your data, and by clicking these different tools, and you get your results. So it's something that's worth trying if you want to play with your data. But again, we have the expertise to take your data to the next level and try to identify the detailed and nuanced information in the data set that probably buried deep in the data set. Another example is a work by Dr. Veera, is the SpaceX, which is a new method for co-expression network estimation using spatial transcriptomics data. The idea is we take this spatial transcriptomics and try to identify this co-expression network, both at the local level and at the global level. So these genes may have different interaction patterns in local regions, because we have this resolution to look at the data at the these different tissues or different spots. But at the same time, if you look at the overall slide of cells, there may be some shared interaction patterns. So this method is powerful enough to dissect those different co-expression patterns by taking into account the spatial information of the transcriptomics. So these are just the overview of some examples, and we have much more methods that are in development, and also we welcome any new questions and new data sets that we can help you with. So I'll pass back to Veera for rest of the talk.
Yeah, thanks, Ken. So I just want to make one point, like, as you might be getting a flavor of this, like we are sort of a mixture core, like in terms of a lot of the faculty who are associated with cancer data science have their own research programs, cross-fertilizing with different groups, research groups here in terms of working with domain scientists, biologists, translational scientists, and clinicians as well. And what we hope to achieve is using those tools, that modern tools that has been in development, and deploy them, across multiple data sets, and bring out the, different aspects of the analysis. So this is one such example that actually my group works on, is on multiplex imaging. As I mentioned, on multiplex proteomics, spatial proteomics, that is been a sort of a recent endeavor over the, I want to say, last 12-18 months. That includes multiple things in terms of pre-processing and all the way up to analysis, and the different flavors of analysis. And some are first order exploratory analysis, which might be just, looking at prevalence of cell types. But going beyond that, looking at second order, which is much more going deeper into cell-cell co-localization and interactions between cells. And not only just doing exploratory analysis, but downstream going into something of a confirmatory analysis by layering that with some level of either outcome data or patient level metadata. So how to do those interactions. So this is a recent endeavor that we took on, stemming from an internal collaboration here at Michigan, of analyzing these data sets as spatial, as manifestations of some spatial processes, and looking at different aspects of co-localization and putting that in the context of clinical modeling. And the part of this, actually, this project was like, what would be some of the accurate metrics? What are the quantifications of cellular co-localization can we look at for multiplex imaging data? It turns out you can't pick one from any of the spatial metrics that are out there, but it needs a little bit more careful thought in quantifying these. So we also, in terms of dissemination, we also have developed as part of this and multiple other projects, R packages, as well as Shiny apps that one can use to implement these methods. So finally, just listing out different types of methods that we provide. We do some level of data pre-processing. We are more at the downstream end of things, but we do some level of data pre-processing, visualization, as Gen mentioned, statistical analysis, more nuanced statistical analysis, especially uncertainty quantification, and more hypothesis-driven questions. And if something is, an off-the-shelf tool can't identify, then we go a little deeper, which becomes much more of a research developmental project, doing some level of methods innovation. So if you want to get in touch with us, please do. We're happy to answer any questions, and happy to hear from any or all of you. Thank you.
Thank you very much. This is a good presentation. So my question is for I see like a lot of tools. Have you done any kind of like benchmarking? Probably like some of the tools they work for some issue, the other for other issue. So I like some of the examples you give us, but have you done any like benchmarking? Do you have any recommendation which tools they work for some issue, other?
I can take that. I can give you one specific example. I can't speak broad strokes, but I can give you one specific example of quantification of cellular co-localization using multiplex imaging data. So multiplex imaging data, especially the manifestation of the data, has a lot of, I wouldn't say issues, but caveats, especially in terms of holes in them, So what is truly a spatial interaction, quantification of that? And that's something we've been thinking about a lot, like what are the metrics? Can we just pick up any spatial metrics that's out there that's been developed, I don't know, 20, 30 years ago and just put it, but it requires a little bit more careful. So that's why we came up with what is called a distance matrix, which is much more, and we calibrated it. There's multiple ways to validate it, but we do it via, some synthetic evaluations as well as looking at what it actually captures. But that's one example now of doing some level of benchmarking. So we do give it some thought. I don't know if that answers your question. I'm just giving you one example.
Thank you.
Justin.
So we're going to be going to a break. I think